This is again the gastroesophageal junction, where we can actually see the difference in the epithelium. We're going to discuss about the epithelium of the gastromucosa just in a bit. So this actually terminates our conversation about our discussion about esophagus and initiates the one with the stomach. Now the stomach is made out of 4 different areas: the cardia of the stomach, the fundus, the body, and the pylorus. Now it is very important to understand that these 4 areas do not have the exact same function. We're going to distinguish areas that produce HCl along with other contents. We'll see what other contents, and areas that produce mucus, specifically the cardia and the pylorus, so the beginning and the end of the stomach, do not produce HCl, do not produce hydrochloric acid, along with, of course, other contents. Now the fundus and the body are the locations of the stomach that produce the, let's say, the main functions of the main enzyme, the main, let's say, components that we care about the stomach. So this is something to bear in mind. First, we'll talk about all the different cells we're going to see in the stomach, and then depending on the location, you will understand yourself where we find what cell. So this is again the gastric pit. The gastric pit is the invagination of the whole layer of the let's say of the columnar cells and cuboidal cells that we find. We're going to talk about these cells right now here. So in the upper part of the gastric pit, we're going to find the surface mucous cell. These are cells that produce alkaline fluid containing mucin. Mucin is just another term for glycoproteins and rich saccharidic content. So this is the surface mucous cell. Exactly beneath and interspersed with them, we're going to find mucous neck cells. These are again cells that produce mucus, but the difference is that this is an acidic fluid containing mucin. Now, exactly beneath the top part, the surface part of the gastric pit, we're going to find the isthmus, the neck of the gastric pit. This is a very, very important structure, because here, in fact, we'll find the stem cells, the precursor cells of all the differences that we're going to find inside the gastric pits. So, amongst, of course, the stem cells, we're going to find 3 different other types of cells: the parietal cell, the chief cell, and the G cell. These actually are going to be found a bit lower in the gastric gland part. Specifically, we're going to talk about the functions of all these 3 cells. First, just namely, and then we're going to visualize again these cells and to actually really go into detail to what they can do. So the parietal cell is the one that produces and secretes hydrochloric acid. The term here, produce hydrochloric acid, should be not used, because we'll see why. So usually prefer the term secrete hydrochloric acid. And of course, also produce intrinsic factor. The function of the intrinsic factor is to, of course, to assist the absorption of vitamin B12. Now, we'll see, of course, how it looks like in just a bit. In this point, just let's focus on the chief cell and G cell, and go back to the parietal cell as to how it does these unique functions. Then we're going to find this highly productive chief cell. This produces, of course, trypsinogen. What is trypsinogen? Trypsinogen is a structure that is an enzyme that will eventually convert into pepsin, and pepsin, of course, is a protease. This is specifically an enzyme that cleaves proteins. And lastly, we're going to find the G cell. Now, the G cell is the very first example of a very, very big family of cells. Specifically, this family is going to be called diffuse neuroendocrine system, DNES, DNES. Now, the DNES is actually a very, very wide range of cells that produce hormones specific that allow to control specific locations of the GIT. We'll see, for example, let's check out just a very few examples of the DNES. Now, there are 2 different types of dead DNES cells: the open cell and the closed cell. Just because they are hormonal cells, of course, this means that they have a very, very close contact with the vascular elements of the tissue. Now, the open cell has a connection to both the local vasculature and, of course, the lumen of the cells, the lumen of the location of the organ as well. The closed has only connection to the vascular location, as we can actually see by the picture. This is an example of how many cells actually can be classified as a DNES. This is, of course, a table from Janquera, and you can actually see the many different types of cells that they have. Typically speaking, the cells are named with a specific capital letter, and again, this capital letter is typically used typically taken from the hormone that they produce. Specifically, the G cell produces gastrin. Now, the gastrin is a hormone that induces the gastric acid secretion, and also it has a trophic effect on the gastric mucosa. You'll see way more of these G cells along the line. It's not very important to remember by heart, just know some more specific hormones you should discuss in histology, and you will focus on these structures and these hormones more in physiology to find out the different interactions between the different hormones that control the local, let's say, autonomic system of the stomach and the GIT. So let's focus just a bit on the parietal cell. I told you before that the using term produce HCl is not really correct. Why is that? Well, because the function of the parietal cell is again a function of ports, a function of channels. The function of the parietal cell is to simply import, using the different channels to import H protons and Cl and chloride, of course, ions. Now, as long as it actually uses these ports to pass these two different components, the HCl, the H and the Cl, in the lumen, these H protons and the chlorine ions are going to be connected in the lumen to form the HCl. So the HCl is technically not produced inside the parietal cell, but is formed inside the lumen. So this cell is actually performs a dual function. First, it produces a very potent acid, highly corrosive to all the tissues. So it secretes this very corrosive acid on the one end. But if this was the only function of the cell, then it would be actually harming itself in a way, because all cells eventually have a specific pH limit they can survive in. So in order to protect the rest of the tissue from this highly corrosive agent, they form the bicarbonate buffer in the basement, close to the basal surface of this cell. And this function is performed by a very important enzyme, the carbonic anhydrase. Now, this enzyme is an enzyme that reacts and plays with the molecules of carbon dioxide and bicarbonate buffers. Using CO2 and OH molecules, it can form an HCO3-. Now, the very important component, the very important, let's say, property of the HCO3 is that because it actually ends up becoming a buffer, it has a specific potential to maintain the pH in a specific range. Of course, it's not, again, a basic pH, but nonetheless, the acidic one that is formed by the HCO3 is way higher than the one of the HCl, meaning that it's way less corrosive, and that it can actually support and stabilize the pH in the basal area of the parietal cell. So I told you so far that this cell has many ports, many pumps that allow for these molecules and these substances to form. The thing is that all of these pumps require ATP, and this is why we see that the parietal cell is filled, and literally filled, with mitochondria, because we have a very high requirement for ATP, simply because of the high amount of presence of the pumps throughout the whole cell. One more feature that we should actually discuss is the intracellular canaliculi. What are these? These are specific connections between the two feet, let's call them feet for now. These two processes of the cellular process of the parietal cell, they actually start to connect and to form bridges from one end to the very other end. Now, why would they do this? Now, the cell, of course, uses ATP, uses energy, uses time, uses cytoskeletal parts to just form bridges. Why is that? Well, because we already discussed this whole logic of the human body to increase surface area. The thing is that because we can increase surface area, we can increase the amount of channels that are present throughout the whole cytoplasmic membrane, and as a consequence, we allow for a way higher amount of these molecules, the HCl intrinsic factor, to being produced and to be secreted in the lumen. So in fact, based on the amount of the intracellular canaliculi, we can assess if this parietal cell is highly active or not highly active. This is again because we have an increased surface area that can support way more pumps to form again all of these chemical molecules, for example the HCl. Now let's talk a bit about the chief cell. Now the chief cell, as I said before, is a highly productive cell. In fact, this has a very granular appearance. Now the chief cell produces pepsinogen, which in the end will be converted to pepsin and gastric lipase. Important to notice that so far we've seen two digestive enzymes in the oral cavity. We've seen the alpha amylase, which already cleaves and of course starch initiates the degradation of starch. Again, starch is a polymer of glucose monomers. And of course we also saw the lingual lipase being produced by the tongue. So these two molecules are already so far being produced in the oral cavity. And now in the stomach we find the pepsin, which is a specific specific cleaves proteins, and the gastric lipase, which also cleaves lipids. So the chief cell is the cell that is responsible for the production of these two molecules, the pepsinogen and the gastric lipase. So this is again the activation. This is a very nice table. This is by the Physiology of 6th edition of Lido Costanzo. Generally, the book of this specific book is, according to in my belief, one of the best books that I've seen in physiology. It actually helps you understand many things with really very well established methods. And for me, that was the one that I used to understand physiology. And from this part, I'm just going to using this table to show you that this is the first actually protease that we see in the stomach, and the low pH is the one that activates and converts the pepsinogen to pepsin. We're going to come back to these tables in the end of the GIT to summarize and do a whole review of digestion. So at this point, just understand what I'm saying. Do not remember pepsinogen and all the molecules that we're going to see and all the specific proteins that we see, the metabolic proteins that we see. We already discussed about NES. And now let's visualize the structure in the macroscope. So let's go to the cardia. This is the cardia of the esophagus. This is the cardiac stomach. So we're going to zoom in and actually visualize what. So as I said before, the cardia does not produce HCl, and we're not going to find here at all not one parietal cell and not even one chief cell. So in this case, we're going to find in the gastric glands and the cardiac glands only specifically these mucus producing cells, the surface mucus cell and then neck mucus. So let's visualize the structures. So in the first part, exactly after the esophagus, we're going to find the cardiac stomach. Now if we zoom in, we're actually going to see only two different population of cells here. Three, sorry. We're going to find the surface neck cell, the surface mucus cells, and the neck cells. Also, the last one is going to be the stem cell, which is going to be found in the isthmus of the gastric pit. So again, if we zoom in, we're going to see all of these cells, which are typically the shape is low-columnar to cuboidal, and along the whole gastric gland portion, we're going to find nothing more than just this mucin cells, the cells that produce mucus, and that's it. So this is the whole mucosa. Again, the submucosa, we're going to find nothing more than just connective tissue, and then again the typical muscularis externa. Again, here we're going to find three different layers. The oblique is the first, then circular, then longitudinal. This is going to be, sometimes we're going to also see the oblique sections, and typically it's going to be found in the first layer before the circular layer of the muscularis mucosa. And lastly, we're going to see, this is very easy to identify, this is the typical adventitia, because we can actually see lots of adipocytes present in this area. In comparison, this is, for example, the serosa. Again, this very nice thin line along the whole line of the esophagus, and this is actually covered with the mesothelial cells. For example, this is exactly the right one. There we go. This is the mesothelial cells right here, along with the line, this simple squamous epithelium. So let's move on to the fundus. Now the fundus, of course, is the area we're going to actually find the parietal cell and the chief cells. How do we distinguish the two? First, let's zoom in. First, let's actually see the layers. This is again the mucosa layer, submucosa layer, and the muscularis mucosa. And lastly, the do we have anything here? No, it's not here. Especially nothing. There's no serosa adventitia here. So let's skip that. So we can actually zoom in first the mucosa layer, and again we're gonna find on the top always we're gonna find on the top the surface mucus cells and the mucous neck cells. This is the typical surface mucus cells. These let's say these folds are also called rugae. So along the rugae we're gonna find this surface mucus cell, and deeper we're gonna find the different types of two different populations, the parietal cells and the chief cells. I already told you that the actively producing cell is the chief cell, and the one that actually does not produce is the parietal cell, because the parietal cell does not produce anything. It simply moves, let's say, ions from one end to the other. So the productive cells are typically the basophilic cells. So if we zoom in the gastric glands, we can actually find out the two different populations of the cells. This, for example, this is the very granular appearance. This is the basophilic one that we can actually easily understand that this is the chief cell, and this is the very, very big and very wide parietal cell. Again, this is a more typical picture of the chief cell. We can actually see also what the author uses. This is the parietal cell again. This is the very, very nice clean section of the parietal cell, and the chief cell is the very basis with a granular appearance. So here in this section we can find both of these cells, the parietal cell and the chief cell as it is. So let's move on. Now, the pylorus, again, this is exactly identical structure when it comes to cardia. Again, we're going to find mucus producing cells, again the surface mucus, and then neck cells. And in the gastric glands, we're going to find only producing of mucus contents. So let's move on to the small intestine.